the panel is at least open to a couple of questions. Please, go for it. Yeah.
For Dr. Patel, any of those future, I saw you had the date on there, February 2017, are you seeing people now to potentially enroll them for next year, or no? Do you want to wait until?
We can't, unfortunately, you can't until it's gone through the regulatory process. If somebody just wants to come and hear what the options are, maybe you don't need a therapy now, yes, we can do that. But if you needed a therapy now, it's better to try one of the ones that's already open, and we can keep those others in mind if we need it when they open.
What are y'all's thoughts on the class 2 patients wanting to think about boric acid? It little side effects and may be beneficial.
I wouldn't recommend doing that outside of Dr. Sato's clinical trial. I mean, his is the only clinical trial in the US or in the world right now studying that in a formal mechanism. So otherwise, giving valproic acid outside that is challenging to know if it's going to work for one person or for the group as a whole.
Yeah, that's a very easy medicine. I know this medicine from my residency because it's a seizure medicine, so most doctor knows how to do it. But what we don't know is whether that is worth doing. So in theory, a very excellent Dr. Harvard's research study might work, but nobody knows actual data. So that's the reason why we decide we are forced to, we have to do some prospective clinical study to show the any efficacy, potential efficacy. And then now we don't have any placebo arm, so therefore we are going to be criticized. But for this specific trial, one of the major purposes is to try the efficacy of valproic acid, so therefore I decided not to put the sugar pill arm. So we will compare the student out, our number is not big enough. At least we can see some parallel, survival, like a survival or parallel time-free progression-free survival. That indicates that maybe these two medicines may be similar. On the other hand, if we see the difference, we might go into the maybe winner is Sutent, winner maybe valproic acid, and then next stage is a painful placebo study.
Now, boric acid has any significant adverse effects?
Yes. The major side effect that you need to be careful is suicide attempt. There is a box warning, and a patient, again, we have one patient who couldn't tolerate 750. 750 is low dose though. We can't give the dose that he's giving to the mice. So he decided to give a highest dose transiently at night. Anyway, even box warning, you can read through very carefully. Suicide thought, suicide attempt is one of the major warning in the prescription, information. Okay, and then more frequently seen toxicity is liver toxicity, platelet decrease, and then dizziness, sleepiness, and then character change.
If you can only get a trial approved with a placebo, is there a point if the success of the trial is so different that it's going to provide a crossover so everyone can go on to the real drug?
I want to repeat the question.
So the question is if the necessary evil is to compare to placebo, would patients who are ever on a placebo maybe get to cross over and actually get the drug? So the short answer is yes. Most phase 3 trials, or some I should say, will have a crossover effect, but the problem is that waters down if you're looking for, say, survival, then the FDA, this happened with Delcath, of course, when they initially went, when the patients who got randomized to best alternative care progressed and crossed over onto Delcath, now the survivals can become difficult to interpret, because now if they look similar, is it because the other group was rescued by the Delcath? So it depends on your endpoint, but these trials are also designed such that they have an interim analysis for futility. So if one arm is strongly outperforming the other one, that will get shut down so that the treatment arm can stay open, because it will seem inhuman to keep a placebo arm open. And some trials right now in skin melanoma are being approved, drugs are being approved without comparison to the gold standard. If that ever happened in a rare cancer like this, if we had a drug that was overwhelmingly working for the majority of patients, even before comparing to a gold standard, the FDA might consider approval. That approval can always be taken away, though, if future trials do not demonstrate a benefit over a gold standard.
I just want to make sure I didn't miss something, but was there any mention of any study that combined an immunoembolization with a chemoembolization? And if not, was there anything in the thinking nowadays that leans in that direction?
Sure. Again, that kind of motivation to move to phase 3 is based on the funding. So, we internally, we decide to the order of the treatment that I'm going to explain that. And then that is eventually have to do, but we are not doing it right now, because the GM-CSF is, company is not interested in doing phase 3 right now.
There actually is a rationale for combining those therapies. When you do a chemoembolization, you increase the inflammatory cells into the liver. That's been shown either with ablation or chemoembolization. If you follow that with immunotherapy, it makes sense you may get a greater effect, additive effect. So there is some rationale behind the combination.
Any other questions?
Just a commercial question. Where does your funding come from? Is it federal funding, or is it us that provide money, or where does your funding come from?
So for some of my studies, I have NIH funding for a few of my studies. The Glemba study is funded by the NIH. I have a separate R21 individual grant that supports the Nevo-IPI study, because that study has an amendment that will require mandatory biopsies in the first 3 months. So I have grant funding to support that. The engineered T cells is supported by the MD Anderson Moonshot platform. BET inhibitor and PV10 and the microRNA are industry sponsored, pharmaceutical sponsored. I don't have any crowdsourced studies right now. I don't, if that's what you're asking.
So there is a 4 source of funding. One, commercial company, they have their own interest in pushing it. But if we will try to make them convinced, we have to show our basic research data. So this is a rare disease, they are not interested in basically. So we spend a lot of time in generating some preclinical data to make them convinced that they can support the clinical trial. And the second is, she has been very successful, public funding. NIH, CTEP is one of the mechanisms that they can give us a medicine and with some funding. So that, again, time consuming, and then you have to show a lot of kind of effort. And number 3 is, patient donation. Actually, Sudan trial, medicine, sunitinib was given by the Pfizer, but we are buying valproic acid and then paying nurse time by the donation from patient. So that's very appreciated. Number 4 is institutional. So if institutions have some motivation to promote, investigate the initial clinical trial, they can support the nursing fee, research nurse fee, but still we have to get the medicine from somewhere. That's the kind of reality. Dr. Feun.
Our trial is supported by a drug company, Novartis, providing the drug and providing the support for the any biopsies and laboratory tests. Also, I mentioned that we're part of the MATCH program, you may have heard about, NCI program, where patients who have tissue available for DNA analysis, if they're eligible, they get a free DNA analysis mutation study, and then we try to match them to any trial or drug they may be qualified for. At least they may be targeted for, depending upon DNA studies.
Everyone understand MATCH trial? It's not much. You want to explain?
MATCH.
This is like removal of the boundary of individual cancer. They are going to make a decision for medicine based on molecular signature. For example, BRAF is mutated, they have a medicine available. For example, HER2 is expressed, they have a medicine. Doesn't matter pancreatic cancer, breast cancer, melanoma. So this is a new trend. So there's many so-called match trial, matching the molecular fingerprint with the medicine. So that is most of the time is a phase 1 or phase 2.
They have 26 different sub-protocols in the MATCH trial. And the MATCH trial is a good resource, but it's also underfunded. So there are trials that we have full funding for, and there are trials that we have very borderline funding, in which we actually have to bring in resources from the institution because the trial doesn't pay for all it costs. One example is the adjuvant crizotinib trial that we're talking about. We think it's important. We get our funding from Melanoma Research Fund, which was created years ago from big donation actually from a patient that has supported a lot of, and we use those funds to support a lot of these borderline funded ideas.
Have you guys, with the MATCH trials, successfully matched a uveal melanoma patient though? That's the problem, is that the mutations that are carried are these GNAQ, GNA11 predominantly, for which there is no, drug out there. So I actually get frustrated with my phase 1 colleagues at MD Anderson when they say, before we put them on BET, we're going to run the match, because that's a 3-week wait. You have to get the tumor biopsy, send it, wait for the results. And I tell them, I'll tell you what the genetic profile will say. It will not match them to anything. Let's just move on. So that's my only caveat.
Actually, every day is frustration. Some match trial give us the opportunity for mechanist. Mechanist 0% response rate. So as a single agent, I don't want to do it. In combination, it's a different story. But,
Are there any further questions? Thank you to the panel again.